Today we'll be looking at our fourth organic macromolecule, and that's going to be proteins. We're going to be looking at how they are built, and that's really what we're going to focus on today. That's our main objective. We are going to talk about a few examples about them, and we'll later on explore functions of proteins. The first images you're seeing here are some protein renderings, some computer-generated images of proteins, and of how they would look based on their shape. And we're going to learn today that shape is very important to the protein structure. And so starting off with proteins, we want to look at what actually makes up a protein. And I have a couple of things going on here, so you might want to take a minute. But proteins are going to be made up of CNOPs. Those are all the possible elements. Some proteins will have that sulfur, but not necessarily all of them. Okay, so that's why we have that possible there. If we look at this first image over here, I have a couple of parts that we are separating for you. Proteins are going to be made up of the monomer amino acid. Okay? And each amino acid, this is an entire amino acid, so 2 amino acids here, and each of them are going to have 3 parts that are similar or are the same. The first of those parts is going to be called our central carbon. Okay? Our central carbon is basically the carbon that all these other functional groups are attached to. Next, they're going to have an amine group. And we want to remember that this is one of those functional groups that are slightly polar. Just like with our other macromolecules, the charges, the electronegativity, the bonding between the atoms are going to affect the overall function or shape of that structure. So the third group that we have here is the carboxyl group, and sometimes it's also known as the carboxylic acid. And so that gives us the acid in amino acids. So amino acid, okay? And there's also a little hydrogen that's always attached to that central carbon, because carbon, again, because of its electronegativity, wants to form those 4 bonds. And those 4 bonds are going to be covalent. Now the last structure, last part of the amino acid, this 4th one, is known as the R group. So we'll write an R group. And it is this variable side chain, and this is what's going to create the difference between amino acids. So just like how nucleic acids or nucleotides, there was A, T, C, and G, and even sometimes U, the R group is what's creating the difference there. What we want to look at is what or how these can join together. So very, very basic, just like our other 3 macromolecules. 2 amino acids can join together by dehydration synthesis. Basically, the hydroxyl group right here, the OH group, and the hydrogen on this side will join together, will join together to form water. Water will be released, and will actually end up creating a peptide bond between the 2 amino acids. And so that's why you get the term polypeptide chain, because of this peptide bond. Okay? Now, what we want to look at next is how are polypeptides, or how are proteins named. Proteins are going to be called polypeptide chains or amino acid chains until they fold up to become a protein. A protein has to have a 3D shape in order to function. If something happens to that shape, then it's no longer going to function. So here we actually see in this image 3 different amino acids. Notice that they all have that amine group, that carboxyl group, and between them you can see that water had been lost at one point, and to get rid of that OH and H, just like up here. And then up top, you have those variable R groups, which are also sometimes known as side chains. So where the central carbon is, where those three essential parts are, that's known as the backbone, the base of the polypeptide chain or of the protein. And then our R groups are actually going to be what causes the interactions that cause the folding or the shape of the protein. So again, the side chains and their properties, their chemical properties and electronegativity and polarity are what's going to cause these interactions that's going to cause them to fold. Okay? So the next portion here we're looking at we're actually looking at 20 common amino acids. Between the 20 common amino acids, we do have more than that, but these are the ones that are necessary to all life. Between the 20 common amino acids, we have three groups: our nonpolar group, our polar group, and our electrically charged group. Okay? If you notice, between each of them, you have those three basic parts: central carbon, carboxyl, and the amine group. But then you can also see the little side chains, and they do vary. The nonpolar group is going to be hydrophobic, and this is going to be important later to their structure as well. And the polar and electrically charged is going to be hydrophilic, water-loving, water-fearing, and that's going to affect their shape because amino acids generally are immersed in water, okay? So these 20 amino acids are going to have different properties that are going to depend on their electronegativity and the bonding between the atoms on the individual amino acid. What we're going to look at next is creating this 3D structure. I keep telling you that the charges and the polarity and the bonding are affecting the shape. And so the shape is very, very important, and is very important to the function of the amino acid. Without that specific shape, it would not be able to do its specific job. Okay? So in biology, one thing to really remember is that form is equal to function. Okay? And so when we're talking about the shape of a polypeptide, we actually look at 4 different levels of that structure. The first structure we're going to look at is the primary structure, and this is actually going to be related partially to those peptide bonds that are created between the individual amino acids. And here you see glu, phi, gly, and these are going to be the abbreviations for those amino acids that we saw above. So we use a 3-letter abbreviation for them. The peptide bonds are very strong bonds. They're covalent bonds. And when we look at the primary structure, we're looking at the amino acid sequence. So for primary structure, 1 degree, this is, again, primary structure. And this is going to be caused or determined by the amino acid sequence. And we should remember that the amino acid sequence is determined by our DNA sequence. The code from the DNA codes for amino acids, which in turn lead us to our protein. The secondary structure is going to be caused by hydrogen bonding. Okay? And the hydrogen bonding is going to come between those atoms, and because of those R groups. The hydrogen bonding is what's going to cause what is known as alpha helixes and beta sheets. Your beta sheets are where you see folding, and your alpha helixes are going to be where you see these spiral structures. But this gives us our secondary shape. Okay, and these are just between the amino acids themselves. So we can add that in, between amino acids. Then the next type of shape, or the next structure, is going to be our tertiary structure. And this is actually what creates our 3D structure. So our 3D structure is actually going to be when our whole molecule folds together. And so here you see those alpha helixes and the beta sheets that are folding in on each other. And when they create the 3D shape, this can be the end of our protein folding. This can make our final product. The folding of the whole molecule really is going to depend on the hydrophobic and hydrophilic characteristics. And again, this is due to those R chains that we saw on the amino acids. Okay? So whether or not it wants to fold in, hydrophobic, away from the water, or out, be on the outside touching the water. Another type of bond that can occur here is also known as the sulfide bonds. If you have a sulfur in your R group, then this can actually form a bond between another sulfur and create stabilization of the entire molecule. And then the last structure we have is called the quaternary structure, and this is a complex of protein molecules. So what that really means is that for quaternary structure, we have more than one plus one subunits. Okay? So several subunits or several separate proteins. So multiple proteins can form a larger protein complex. An example of this is going to be hemoglobin. Okay? Hemoglobin actually has 4 separate structures, and those 4 separate structures are actually made up of 2 alpha helixes and 2 beta sheets, and they'll fold in on each other because of their chemical nature. But sometimes that shape can be lost, and when that shape is lost, that's called being denatured. So if you lose your shape, and lose is spelt wrong, if you lose your shape, you can no longer function. Now remember that our 3D structure is determined by our hydrogen bonds being hydrophilic or phobic, and just general interactions. Okay? Now you can be denatured or lose your shape because of a change delta in the environment, such as pH, the acidity, the temperature, and the salinity of a solution or of an environment. If the pH changes, remember this is going to be your concentration of hydrogen ions concentration, and this is going to actually change bond interactions. Your temperature, when this changes, you're normally adding heat. This can break bonds. And for salinity, you're adding in salts, so NaCl-. And what this can do is this can actually interrupt bonds, because remember that the bonds, the charges, the polarity, the electronegativity can cause these bonds to form together. And if you have these added in there, it can separate them, or it can block them from forming properly. So when you interrupt these bonds, when you do these things, when you're changing all of this in the environment, you interrupt those bonds, and you cause problems in the primary structure. And so then you can end up with no peptide bonds. You can even break it down all the way to the very beginning, where you'll no longer have your polypeptide sequence. Okay? And all of this will lead to it being non-functional. So you have your folded up protein. If you're being denatured, that can be pH,